Let me now turn to Dr. Jacob Haus from the University of Michigan. And Jacob, please share your screen and take it away, talking about some protein modifications in obesity and diabetes.
Good morning, David. Thank you. Everyone should be able to see my screen now. Is that correct? Good. So, David, thank you for the invitation. It's a pleasure to be part of this Muscle Health Awareness Day. And I just want to say nice job in putting all this together. It's a big task to coordinate these things and put it into a webinar. So nicely done. And I'm looking forward to talking with you about our story that we've been working on for a number of years now. So the main focus of my laboratory is diabetes complications. And specifically, we're interested in the exercise biochemistry and the skeletal muscle health as it pertains to diabetes complications. So you can imagine here that skeletal muscle would play a big role in, say, peripheral artery disease, peripheral neuropathy, and even in the diabetic foot. And what's not shown here on the this wheel of complications is also cognitive decline, as well as diabetes-induced myalgia or diabetes-induced muscle wasting. And so we think that exercise biochemistry has a lot to play in those particular spaces. Now, when it comes to understanding diabetes complications, we've typically been focusing on glycation, oxidation, and nitration-induced protein damage. And so we believe that these cumulative or long-term changes in stable macromolecules lead to tissue damage, and that hyperglycemia, of course, is a major driver of that. And then, of course, you can see how this is a vicious cycle in terms of damaging the tissue and superimposing genetic determinants and comorbidities such as hypertension and hyperlipidemia. But we and many others believe that these glycation, oxidation, and nitration-induced protein damage is a major driving mechanistic force for the development of future complications. So with regard to glycation, we typically associate reactive dicarbonyls in glycation biology. And so when we form a reactive dicarbonyl, you can see the structure here of these dicarbonyls on this right side of the panel here. You see methylglyoxal, glyoxal, and then 3-deoxyglucosone. And you can clearly see their dicarbonyl side chains here. These are what are reactive with the protein groups. All of these molecules can rapidly glycate and damage proteins, and we know that they primarily interact with arginine residues. Now, we're not alone in understanding these types of complications or focusing on it, especially with regard to skeletal muscle health. And so here's a series of large-scale clinical trials that have involved muscle strength and health in older adults, as well as those with just existing comorbidities. The Incanti study is a very famous one in terms of showing that advanced glycated end products were associated with decreased mobility and decreased lifespan in this particular study. And then also the accumulation of these advanced glycated end products in the skin and the muscle also predict mortality in a study here published in 2011. And so we think it's important to continue to focus on the skeletal muscle with relation to glycation biology to not only understand age-related processes, but also to help gain insight into diabetic-related problems as well. Now, glycation typically affects arginine residues. And so arginine is very prevalent in catalytic domains. And so here you can see some data published a number of years ago now from Bartlett that shows that the percentage of arginine residues in catalytic domains is actually quite high relative to histidine and asparagine. And so if we look at a more specific example, here we see the muscle-specific isoform of pyruvate kinase. And what's been identified by proteomics work is that a very critical arginine residue in the ATP binding domain, Arginine 120, was shown to be modified in this muscle-specific pyruvate kinase in mice, and it found to be negatively associated with determinants of metabolic health, as well as some in vitro work to show that the pyruvate kinase activity was negatively affected by this particular modification. Now, here's some more direct skeletal muscle functional type of analysis. This is some data that was published in Diabetes now over 10 years ago, but it shows that when you incubate L6 myotubes with increasing concentrations of methylglyoxal, you see that the insulin-stimulated 2-deoxyglucose uptake is proportionally reduced as you increase the amount of methylglyoxal stress onto the L6 cells. And that this effect could be rescued by using a methylglyoxal scavenger known as aminoguanidine. Now, again, methylglyoxal is formed in proportion to glycolytic flux, and it can make up around anywhere between 0.1 to 1% of all of the glucotrioses that flux through glycolysis. Now, importantly, it's the 3 carbon intermediates, the G3P and the DHAP, that are responsible for the formation of methylglyoxal. What happens is they spontaneously degrade into a methylglyoxal product, and this just happens spontaneously based upon the conditions inside the cell. This happens in normal people anywhere between 0.1 and 1%. However, if you have a diabetic condition, this can be elevated up to 10 and sometimes in some cases even 20-fold if the hyperglycemia is uncontrolled. Now, when methylglyoxal flux increases, we have that likelihood that it will interact with a protein side chain, again, more specifically an arginine residue. So again, you can see here the dicarbonyl side chains have an affinity for the arginine residues, and they form now a intermediate product called an MGH1 or hydroimidazolone. This is considered to be an advanced glycated end product. Once this modification occurs, the protein function structure becomes changed. Now, thankfully, biology has a system for the detoxification of methylglyoxal, and that is through the glyoxalase enzymatic system. And how the glyoxalase enzymatic system works is that methylglyoxal is combined with glutathione, and glyoxalase 1 or GLO1 forms that into SD-lactoglutathione, an inert product. SD-lactoglutathione is then picked up by glyoxalase 2 or GLO2 combined with water, and then it's converted into D-lactate, and then the glutathione is recycled. And so you can imagine a condition when we have a tip in the scale towards glycation and decreased detoxification, that diabetic complications would become more prevalent. Now, in terms of the tissue specificity of methylglyoxal production, the liver seems to be the most prominent producer of methylglyoxal, and the advanced glycated end product MGH1, which is, again, this residue that has been modified by methylglyoxal. Now, skeletal muscle comes in a close second. You can see here that proportionally, skeletal muscle has a significant amount of free methylglyoxal, as well as the amount of MGH1 residues, which is expressed in millimole per mole arginine. Now, our first entryway into this subject, so to speak, was a number of years ago. It was led by a PhD student at the time, Jacob May. And so Jake had the idea that, of course, that GLO1 would be reduced in diabetic skeletal muscle. And so that was the focus of his dissertation. And this is, of course, exactly what he saw. Compared to young, lean, healthy controls, which was used as a marker of optimal health, compared to the diabetic subjects, we see approximately 75% reduction in the amount of GLO1 protein. And then interestingly, we saw that GLO1 protein was positively correlated with the amount of insulin sensitivity as determined by a clamp-derived glucose disposal rate, the hyperinsulinemic euglycemic clamp, which is the gold standard for assessing insulin sensitivity. The higher amount of GLO1 expression you have, the more likely the individual was to have higher amounts of insulin sensitivity. Now, inverse to that, the higher amounts of BMI resulted in lower amounts of GLO1 protein. And so this was very interesting to us. And so we wanted to understand, well, what is the mechanism of why GLO1 protein is reduced in type 2 diabetic skeletal muscle? And so we thought, okay, GLO1 protein is down. Well, we have to first look at the transcriptional regulation. So how is GLO1 transcriptionally regulated? Well, it's regulated by NRF2. And NRF2, as many of you know, is an important transcription factor responsible for the transcriptional regulation of antioxidant and anti-inflammatory genes. So the way that NRF2 works is that it is sequestered or chaperoned by a protein called KEAP1. And when KEAP1 is around, it will bind NRF2, and then it will allow NRF2 to be tagged by ubiquitin. And then, of course, ubiquitin is recognized by the proteasome, and then NRF2 is degraded. However, under oxidative stress or conditions when electrophiles are present, critical cysteines on KEAP1 become oxidized, and then KEAP1 will disassociate with NRF2, allowing NRF2 then to translocate to the nucleus where it can bind to its antioxidant response elements and then transcribe critical proteins such as GLO1. And so we examined NRF2 protein expression in our healthy controls and type 2 diabetic subjects, and we found that NRF2 was markedly reduced. And the inverse of that was that KEAP1 was significantly elevated. And so this made a lot of sense to us that GLO1 was dysregulated at the transcriptional level. However, when we went back to our correlation with regard to BMI and GLO1 protein expression, we became intrigued by the idea that obesity alone could perhaps attenuate GLO1 protein expression. And so we embarked upon another study in relatively young, mildly obese, and fit individuals. You can see here that they were age-matched, that the BMI is elevated, consistent with obesity, and that they are relatively fit in terms of their VO2 max. The obese individuals had a VO2 max of 37 mLs per kg per minute. So, nothing to be too concerned about in terms of their low fitness. What we found, of course, was that GLO1 protein was reduced by 50% compared to the lean, healthy controls. And this surprised us, given that they were young and that they were only mildly obese and that we considered them to be relatively fit. And so then what is the consequence of this? Well, these folks are not diabetic yet. They're metabolically healthy with regard to cholesterol and other things. And so we went to the literature and we found some cross-sectional comparisons here is that when an individual becomes overweight, their plasma methylglyoxal levels start to elevate. And we can see about a 46% increase in the amount of plasma methylglyoxal just by being overweight alone. And then with obesity, we see that is about twofold higher than the lean controls. So why does this happen? Well, when we look at this data here by Messon et al. In 2015, we see that during an oral glucose tolerance test, which is 75 grams of oral glucose ingestion, and then glucose values are tracked over a 120-minute period of time, here you see that in normal glucose tolerant, impaired glucose tolerant, and type 2 diabetic individuals, that the amount of glucose in the blood is proportional to the disease state or the pre-diabetes state. But what was interesting to these researchers and us was that during this same time course, you see identical appearance of your dicarbonyl species, methylglyoxal, glyoxalase, and 3-deoxyglucosone into the circulation, proportional to the degree of their disease status. And so these plots mirror one another with regard to the dicarbonyl appearance into the blood. So this again suggests that dicarbonyl formation is proportional to glucose metabolism. And so then we wanted to understand this further in our GLO1 reduced obese subjects. So the next couple panels I'll be showing you here show lean healthy controls and obese subjects again, but we also put them through a acute bout of aerobic exercise. This was 30 minutes of treadmill exercise, and that was performed at 80% of their VO2 peak. And so I want to draw your attention here to the GLO1 transcript. Right, this is GLO1 mRNA that's expressed in copies of nanogram per cDNA. This was done by digital droplet PCR. You can see that GLO1 transcript was markedly elevated in the obese individuals. And so this was surprising to us. This should manifest with increased amounts of GLO1 protein, but it does not. And so we tried to understand, well, okay, if GLO1 transcript is elevated, then NRF2 should also be elevated. And then, so if you draw this attention now to your lower panel, you can see the NRF2, KEEP1, and then with regard to protein, and also NRF2 and KEEP1 gene expression. We don't observe any statistical differences between lean healthy controls and obese individuals with regard to NRF2 or KEEP1, which was contrary to our hypothesis given that GLO1 transcript is elevated. And so what's going on? Well, a new PhD student in my lab, Mr. Edwin Miranda, he thought, well, GLO1 protein reductions could be explained potentially by post-translational modification. And so he thought, well, okay, perhaps GLO1 is becoming modified by acetylation, which would then tag it for degradation by ubiquitin. And so sure enough, when he took some skeletal muscle tissue from muscle biopsy samples we had in these obese individuals, and he performed a immunoprecipitation of acetylated lysine residues and compared to an IgG control, he found that the GLO1 protein is in fact acetylated. And that when we look at and compare it against the IgG control, we find it absent from there. And when we do this experiment between healthy controls and obese individuals, we see about a 13% increase in the amount of acetylated GLO1. Now, Edwin also believed that acetylation may be influencing GLO1 activity. And so he performed some in silico mutations to determine whether or not key residues of arginine in the GLO1 protein would result in potential deleterious enzymatic function. And sure enough, many of these residues would result in a deleterious enzymatic function in an in silico model. And so Edwin then went on to try to elucidate this mechanism a little bit further. And so if we're thinking about acetylation, we have to turn to sirtuins. And so sirtuins have actually been implicated in regulating GLO1 acetylation in proteomic studies. Some proteomic studies have identified a GLO1 residue that is acetylated. And so we capitalized on this idea as well as our own data from human skeletal muscle, with the idea that, of course, exercise and caloric restriction would modulate SIRT, whereas inactivity and obesity would then, of course, perturb the redox state that then downregulates SIRT2 and 1. And then, of course, if we consider NAD biology as well, we capitalize on the idea that NAMT is certainly involved in the synthesis of NMN to create or increase the pool of NAD and activate SIRT1, that nicotinamide riboside is a parallel arm in increasing the total NAD pool, and then also that an inhibitor, FK866, could inhibit NAMT, and so we could then troubleshoot potential regulation in the way that SIRT interfaces with GLO1. And so Edwin went on to probe these samples a little bit further. He found that SIRT1 protein was also reduced in with obesity and that it correlated with GLO1 protein expression. And when we examine NAMT protein expression, we find that there is a trend for NAMT to be reduced with obesity as well. So this would support our hypothesis that SIRT and NAMT or NAD bioavailability may be influencing GLO1 function, thus reducing its content in obese adults. So then Edwin went on and he did some human myotube studies. These were cell culture studies where he treated the myotubes with a NAMT inhibitor, FK866. He also treated the cells with nicotinamide riboside, and then in combination of FK866 with nicotinamide riboside. So if you look at the highlighted panels here, you can see that compared to control cells, treatment with FK866 reduced GLO1 protein, and it increased the amount of acetylated GLO1. Now, if you compare that to the nicotinamide riboside treated cells, we don't see that effect, or we don't see an effect above control in the nicotinamide riboside treated cells. But when we look at FK866 plus nicotinamide riboside, we see that there was a rescue effect compared to FK866 alone. And this was true not only for the GLO1 protein, but also for the acetylated GLO1. Now, we took this a step further and we did some knockdown studies in our human myotubes, and we found that when we knock down NAMT, we see an effect on GLO1 protein. And similarly, if we knock down SIRT1, we also see an effect in reducing GLO1 protein as well. So we think that we have a story here that supports NAD bioavailability, perturbation of sirtuins, possibly through NAMT, that regulates GLO1 through its acetylation. Now, I've gone a bit fast, and I didn't expect to go so fast, but I'm going to wrap things up in terms of conclusions here. So to conclude, GLO1 is a cytoprotective enzyme. It's important for the detoxification of methyl glyoxal. If methyl glyoxal accumulates, then that can negatively affect proteins inside the cell, and it can also accumulate into the circulation, as was shown in some of those clinical studies with diabetes. Now, obesity appears to reduce GLO1 in skeletal muscle, as does frank type 2 diabetes, but the regulation appears to be different. With obesity alone, we see that the transcriptional machinery seems to be intact, but there are post-translational modifications that may be mediated through SIRT1, which is also reduced. And so our in vitro data would suggest that this is an acetylation event, and that would attenuate the protein levels in skeletal muscle. So our next steps in this process is that we've developed a plasmid which has a V5 tagged GLO1 that we've overexpressed now into HEC293 cells, and we're examining these mechanisms a little bit further. Edwin is also doing these same experiments and then taking the samples over to proteomics to see if we can confirm the exact residues that are being modified. And then we'll determine whether or not those specific residues are influencing GLO1 activity. And then also we have a parallel human study going on where we're trying to examine this a bit further and deeper using these same tools that we've kind of discussed here so far. And so I want to acknowledge Edwin here again. This is the focus of his dissertation work. He's been instrumental in organizing and executing all of this, even amidst the COVID crisis. Key collaborators are Dr. Andy Ludlow, who provided the skeletal muscle cells, Dr. David Lombard, who's a sirtuin biologist at UM, and then, of course, Jake May, who is a past lab member, who's now moved on to Pennington Biomedical with John Kerwin. And this funding was supported by Edwin primarily, and also with my current R01. So I'm very happy to answer plenty of questions.
Jacob, thank you so much. That was great. Wonderful, wonderful talk and wonderful explanation of a pathway that we really don't think about very much. And I really appreciate the fact that you're promoting the understanding of this pathway, because I'm not sure there's a great deal of research being done. And is this a big field that perhaps we're missing the significance of? What do you think?
I think it's extremely important. What I don't think it's caught fire, so to speak, is that a lot of these techniques are difficult, especially like examining the glycated residues. They involve mass spectrometry techniques, and there's just not a lot of folks that do it or want to do it. And to be quite honest, it's very difficult. And we've struggled with it in terms of finding an assay that works for identifying these MGH1 residues and other advanced glycated residues. But in relation to diabetes, it's very prominent research and very well acknowledged that advanced glycation is important for the development of future complications. In the exercise world, I think, I may be on an island, but that's okay. I'm happy to be there and
it's
see
unique. It's unique. I mean, it's a unique area. Does exercise itself, speaking of exercise, does acute exercise affect the glycation of some proteins? Have you ever done an acute study to see if the glycation is affected by acute exercise? Just like, glucose metabolism is clearly affected by acute exercise. What about these glycation products? Are they affected by acute exercise?
Yeah, that's a lot of what Edwin is working on right now. We're performing those oral glucose tolerance tests before and after acute exercise, and we're looking to see whether or not the shape of those curves with the appearance of methylglyoxal and such changes with acute exercise. We think it does. We also are taking muscle biopsies, of course, and we'll be looking to see whether or not the protein-bound products of MGH1 are changing with acute exercise. And that's dependent upon the assay, like the proteomics assays and things.
Yeah. Chris Perry asks, are MG and age elevated in other disorders characterized by hyperglycemia, such as cancer or infection? Or in other words, is this a common problem in multiple diseases that we need to appreciate?
Yeah, I think it absolutely is a common problem. It's been implicated in neurodegenerative diseases. It's also been implicated in other neurological disorders such as schizophrenia and anxiety disorders. And so the nerves seem to be particularly sensitive to these glycation events. With regard to cancer, I don't think a lot of it has been studied, but I would imagine that if any hyperglycemia ensues, that you would see curves identical to what was to those observed, say, with obesity, prediabetes, and diabetes. Yep.
Do you recommend pursuing some more NRF2 directed studies to figure out how GLO1 is transcriptionally regulated? Is that NRF2 seems to be involved in all kinds of antioxidant activities? Is that a path that's worth pursuing?
I think it's definitely worth pursuing, but it's a tricky little bugger. And I think those that have embarked into that world know that the antibodies are fickle, that NRF2 kisses and runs, that it runs all around the cell. It dips in and out of the nucleus, and it's hard to catch. And so it's a tricky field, but it's definitely worth pursuing. And, we're not sold on the idea that GLO1 is only dysregulated by post-translational modification. There may be other things that we're missing, like microRNAs, circular RNAs. There's a whole other field that we need to start peeling the layers back to determine whether there are concomitant or parallel mechanisms of dysregulation as well.
Yeah, that's fascinating. Okay, Jacob, thank you very much. That's a great talk and a great educational demonstration of this pathway. Thank you.